Welcome. Welcome. This is the inaugural meeting of what we're calling a conversation about series. But it's not without precedent. It was actually the brainchild of my good friend, Rena Horowitz, who years ago had a group of 12, G12. I thought they said for girls, 12 girls.
12 and friends.
But they're actually a group of 12 and friends, and they wanted to have a community outreach based at Sanford Burnham Prebys to educate us in areas that maybe most of us are not comfortable in, and to bring experts who would calm down a little bit and not overwhelm us, but explain very important topics. And it was unbelievably successful. And then for COVID, we kind of lost some momentum. And then when I gave Brenham moved to Sanford Burnham Prebys, and Rena and I rediscussed this, we said we're going to reinitiate this. We're starting with 4. Rena says she can fill the whole calendar if I let her. But before we go any forward, I want to give Rena a round of applause. And I want to just, so Rena's an old friend. I want to introduce also an old friend, sorry. Not by age, but by how well I know you. So Sandy Larakis was a colleague of mine at UCSD, and then at Salk, and then at UC Riverside, and starting like yesterday, she is now the head of philanthropy at Sanford Burnham Prebys. And she and Rena and also Monica sort of brainchild this, and it's an integral part of what we want to do at Sanford Burnham Prebys. And the underlying hypothesis is that we at this institute have identified several areas that we want to emphasize that we actually think we can translate research into improving health. And one of those areas is cancer, and another one is aging. So it seemed like what a great opportunity to combine them, because virtually every cancer, the risk goes up with age. And so it is a really incredible, opportunity to take advantage of our expertise. And as you'll hear from the speakers, we have colleagues from UCSD, we have new researchers and senior researchers at Sanford Burnham Prebys, and we want to really put together what does the average person, what does an interested person want to know about the relationship between cancer and aging? So we're going to start with a very brief video, and then I am going to turn it over to people who know the field. So I'm going to sit and we're going to play the video. Thank you. There's no place quite like Sanford Burnham Prebys, an independent research institute that for nearly half a century has tackled some of the most pressing threats to our health. This is a time of accelerating and transformational discovery. Our mission is to decipher the most debilitating diseases, creating and advancing effective therapies that benefit patients. Doing so requires people with big ideas and the talent and expertise to make things happen. You can find them here, experts in cancer, genetics, cardiovascular, neurological, and metabolic diseases, immunology, stem cells, and aging, working together and with our many collaborators across the region, country, and world. We also recruit and train researchers in our graduate school and postdoctoral programs. New generations of great thinkers and doers. This is a new golden age of medicine. The old ways of thinking about and doing science are being revised, reinvented, and replaced. Our mission, however, remains the same. We translate science into health. Welcome to Sanford Burnham Prebys.
Okay.
You're up.
Hello.
Hello.
It's such a pleasure for me seeing all our old friends and some new friends. Glad you could be here with us. David said a whole lot of what I was going to say, so I won't repeat it. But I will tell you that one of the new things we're going to do is that we're going to have a study guide that we're going to hand out at each program. It's been a pet project of mine for such a long time because people are using a lot of words that the scientists know about genomics that the rest of us don't recognize, and showing pathways that we don't understand what it's all about. So each time now we'll give you a handbook that's going to talk about the terms, about the presenters, about the studies, and also let you know that we're going to film each session, including the Q&A. So if you've got questions to ask, you should know you'll be recorded. And I have to tell you, already David told you, but our topic is the intersection of aging and cancer. And following our program, there'll be light refreshments outside. You can stay, enjoy some good goodies, and continue whatever conversations interest you. And so now we'll get on to a conversation about, today, a conversation about aging and cancer. And it's my pleasure to introduce our moderator, Dr. Brooke Emmerling. So I'll tell you a little bit about her and then I'll get back out of here. Dr. Emmerling is the director of the Cancer Metabolism and Microenvironment Program and an associate professor at SBP. Dr. Emmerling's research focuses on understanding key signaling and metabolic pathways involved in the regulation of cancer. Currently, a major research project in her lab is determining the role of a family of enzymes that she discovered that are required for the survival of breast cancer cells. And importantly, targeting these enzymes with novel drugs that inhibit breast tumor growth, at least in preclinical models. And so, with great pleasure, come on, Dr. Emmerling.
Thank you, Rena. Good afternoon, everyone, and thank you for joining us for a conversation on aging and cancer. So in your lifetime, you've probably been affected by many people, or at least one person has been diagnosed or has passed away due to cancer. It's the second leading cause in the United States of deaths, and actually every 1 in 2 males here and 1 in 3 women will be directly affected by cancer. So that's pretty strict, right? So cancer's ubiquity is really linked to something a lot of us can't change, is aging, right? And actually we're going to talk about that today, and how aging is really one of the most important factors in most all of cancers. So today we'll be learning about a few of the innovative research projects from our scientists that are leading the effort to understand the links between aging and cancer. And so I encourage you really to jot down some questions, because at the end of this we're going to have a Q&A session with the presenters. So we'll hopefully have some good discussions there. So then, over, like David said, more than 40 years now, it's been exciting that Sanford Burnham Prebys has been at the forefront of cancer research in its efforts to develop new therapeutics to help those affected. The Cancer Center is actually only one of the 7 basic NCI designated cancer centers in the United States. This designation recognizes the Institute's scientific rigor, discoveries, and leadership in finding therapeutic cures. Our researchers collaborate to answer fundamental questions in cancer biology and to help address the critical needs by conducting translational studies. So our Cancer Center has 3 programs that we've highlighted here. One is the Cancer Genome and Epigenetics program that Peter is a part of, that aims to better understand how cell nuclei give rise to tumors and their malignancies. With this knowledge, we learn how to prevent, block, and repair the genetic growth programs that lead to cancer. The Cancer Metabolism and Microenvironment program that I'm a part of seeks to understand how cancer cells survive and thrive in the cancer microenvironment, and really how we can intervene and combat these metabolic pathways in cancer. The Cancer Therapeutic Molecular Therapeutics program uses the most sophisticated technologies to advance the discovery and development of new cancer drugs based on emerging insights into cancer biology at the molecular level, and notably by leveraging the expertise and resources at the Sanford Prebys Center for Chemical Genomics. So our cancer experts specialize in all these programs, but as you'll learn today, we really work together across disciplines with many others throughout the region, country, and even the world to decipher the most debilitating diseases like cancer and create and advance effective therapies to benefit patients. So now, today I'm pleased to introduce Dr. Peter Adams. Dr. Adams is the director of the Cancer Genome and Epigenetics program and a professor at SVP. Dr. Adams' research focuses on epigenetics as a cross-cutting scientific theme and promoting access to single-cell technologies at the Institute. So please welcome Dr. Peter Adams.
Thanks, Brooke, and thank you very much all for coming. It's really great to have the opportunity to talk to you about, well, this really, what I think is a really fascinating, well, important, but also fascinating problem of the relationship between aging and cancer. So before I hand over to Xiao and Kay, I'm going to just introduce the problem a little bit. So first of all, is this working? Just about. So what is aging? So we all, I think, recognize aging when we see it, but as biologists and scientists, what is it? How do we define it? Okay, so aging we characterize as being the loss of function with age, loss of resilience. Okay, so older people tend to be less resilient to physical and emotional stress. Increased risk of frailty, disease, and morbidity, and ultimately, obviously, increased risk of death. Okay, so this is how we characterize aging. Why is aging important? It probably really doesn't need to be said, I think. Brooke has already mentioned this, but aging is the biggest single risk factor for most of the common adult human cancers, and also the biggest single risk factor for all of these diseases that you see here. Type 2 diabetes, heart disease, the biggest killer ahead of cancer, cognitive decline, Alzheimer's, etc. Okay? Aging is the biggest risk factor for all of these. Focusing in on cancer a little bit more, this shows you the incidence of all cancers or specific types of cancer by age. Okay, so you can see here that with increasing age, there is a dramatic increase in the incidence of all of these common adult human cancers. Okay, colon cancer, lung cancer, breast cancer, which is what we're going to focus on today, brain cancer, etc. Okay, so it's because of this, the fact that aging is such a big risk factor for all of these common diseases and causes of death. That's why it's really important to understand the process of aging and how it contributes to these diseases. And so in my lab, we're particularly interested in the role of aging as a cause of cancer. And we're looking at this, a number of different cancers in the lab, but today we're going to talk mostly about breast cancer. So why does the incidence of cancer and also these other diseases, why do they increase with age? So as biologists, we tend to think about what we refer to as the so-called hallmarks of aging. Okay, so these are various different molecular and cellular processes that occur with age, basically accumulation of different types of damage that happen with age. Okay, and you can see here from this cartoon that it affects many different molecules, cells, and tissues within the body. Okay, pretty much anything that you can think of changes with age and accumulates damage with age. Now, I'm not going to go through these in a lot of detail. I will highlight these epigenetic alterations, okay? So this is basically how DNA inside the nucleus is folded up inside the nucleus, okay? And that's something that Xiao is going to talk about later. Another one which is also very important is this so-called genomic instability, okay? And what that basically means is that as we age, the DNA inside the nuclei of our cells, that DNA accumulates mutations, okay? So in other words, it accumulates damage. And that damage, those mutations, can also be, in some cases, can give rise to what we call as oncogenes, okay? And so these are basically the genes, the mutated genes, the damaged genes that promote cancer. And so this week, you may be aware that this is the week where a lot of the Nobel Prizes in medicine or physiology and chemistry have been announced. And so this is a Nobel Prize from 1989 to Michael Bishop and Harold Varmus, and they discovered the role of these mutated oncogenes as important drivers of human cancer. And so now, with this knowledge, okay, we can now draw on this knowledge and try to use this knowledge to understand, well, how does this knowledge of oncogenes, how does that explain the age dependence of human cancer? And so the first thing to note is that our DNA is continually assaulted by different chemicals and toxins which cause these mutations, okay, which give rise to these oncogenes. These might come from, for example, cigarette smoking or ionizing radiation, but some of them just come from the normal physiological processes going on inside the cell, okay, simple metabolic processes. Okay. These also have the capacity to damage DNA and give rise to these oncogenes which cause cancer. And so as all this damage accumulates with age, that contributes to the increased incidence of cancer with age. So in my lab, we've been looking at this more specifically in the role of breast cancer, as I say, and we're using mouse models here, but it's important to emphasize that everything that we do in mice, we also validate in human tissues, okay? We obviously can't do the experiments in humans, but we can confirm that what we find in mice is relevant in human tissues and in human breast cancer. And this is where, obviously, collaborations with Kay, who's here in the audience and will speak in a few minutes, that's where these kinds of collaborations are really important. So most of the work that I'm going to show you here, this is work done by Shanshan Yin, a really fantastic postdoc in the lab. Shanshan is also here in the audience, so you can get a chance to speak to her. But Shanshan has been using this mouse model of breast cancer, and this is a model that was taught to us by Kay, working in Jing Yang's lab at UCSD. And so what Kay and Shanshan have done is basically to induce breast cancer in young mice and old mice by injecting the mammary ducts with these breast cancer oncogenes, and then looking at the frequency of cancer in young mice versus old mice. And all this simply shows is that the old mice get more cancer than the young mice. Okay, so this recapitulates what we see in humans and now gives us a mouse model to be able to investigate this process. And so, to cut a very long story short, I mean, this is several years of work on the part of Shanshan and Kay, but basically what they found is that the old tumours are what we call cold, okay? And that means that the old tumours, the tumours in the old mice, lack the immune cells, which at least in the young tumours, those immune cells will get into the cancer and they will kill the cancer cells. And now for some reason that doesn't happen in the old tumours, okay? And so that's one big finding that we've made, and obviously we're now trying to understand why that is. And actually this now reconnects with the work of Xiao, because it seems that the reason that the old tumours don't recruit all of these immune cells is because of these epigenetic changes, okay? So they have differences in the way the DNA is packaged up inside the nucleus, which changes the cells in such a way that they don't recruit all of these immune cells. Okay, so therefore, in the old mice, the tumours continue to grow. And so, Xiao will tell you more about how we're now exploiting this observation as a collaboration to now hopefully actually prevent the incidence of cancer in the old mice. And so the ultimate goal of what we're trying to do here is to understand why does the incidence of cancer increase with age, and then instead of then trying to cure it, we can actually prevent it from happening in the first place. That's the goal. And so with that, I'm going to hand over to Xiao. Just a brief introduction to Xiao Tian and Kei Yun. So Xiao is an assistant professor here at Sanford Burnham Prebys in the degenerative diseases program. We recruited him from Harvard University last year, and we're already very excited by all the work going on in Xiao's lab and all the exciting collaborations that we have ongoing. Xiao is an expert in this so-called epigenetic reprogramming for the purpose of rejuvenation. And Kei is as I say, we've been collaborating with Kei for many years. Kei is an associate clinical professor at UCSD, a board-certified medical oncologist and a physician scientist specializing in breast cancer. And so you can already see how these kind of multi-component collaborations, including the basic scientists at SBP and the physician scientists at UCSD and other institutions, can really help us to make important discoveries. So with that, I'll hand over to Xiao.
Good afternoon, everyone. Thank you for joining us. My name is Xiao. Thank you, Peter, for the introduction. So, I'm an aging scientist by training. Okay, so I'm very happy to be recruited to SBP to join a collaborative effort to conquer many of the age-related diseases. So, I got interested in studying aging, mostly because, literally aging strikes every one of us. There's no escapers in the face of aging, unfortunately, right? So, including me, I'm losing hair, I'm getting gray hairs. So the problem is, aging has such of a tremendous effect on everyone by inducing all kinds of age-related diseases. So, every family will have members that will be struck by some of these diseases, which is a disaster. I remember that both my grandmas died from cancer, and they suffered a lot, and I'm happy that they don't suffer anymore. And my grandpa died from stroke and heart failure. So that's the thing, right? It's devastating. And my interest in studying aging and the link between aging and the disease is trying to find if we can actually target these diseases by targeting the underlying aging mechanism. Okay, so because it's so common, right? So aging has been considered as a natural process, right? So many of us will think aging is natural. What we can do with it, right? But it may be not true because I'm not sure about the ultimate goal of humans, like what we all design for. But the attempt for human to do is to change things, right? So we cannot naturally fly, but we can fly now. We cannot naturally move at the speed of 90 miles per hour, but that's literally what we are doing every day, right? And also because of that, we got tickets. So, that's the thing. So at SBP, what we try to do is, can we change this natural-looking disaster? Okay. So can we actually apply to what we have learned from the aging research to really bring down the incidence of cancer by targeting the underlying aging mechanism? So I will introduce how my lab study aging. Okay. So my lab study aging from two aspects. So the one aspect is to study aging from the common approach of understanding how aging happens, and on the other side we study how aging does not happen. Okay. Because this is a common approach because to know the disease is required to conquer the disease. Right? We have to know aging to deal with aging. But already there are many long-lived species in nature. After millions of years of evolution, there's some species that actually naturally live long, and they don't develop the age-related diseases that we develop. Okay? So my lab used this two approach to study how aging happens and how aging doesn't happen, and hopefully we can learn and using these mechanisms to target age-related diseases. So from the left-hand approach, we can design interventions to target these mechanisms of aging, okay, with the hope to actually prevent or reduce the incidence of age-related diseases like breast cancer. And on the other hand, we can utilize the mechanism that these longleaf species evolved and translate them into human and see whether we can utilize this mechanism to help us to make ourselves more resistant to age-related diseases. Okay. So today, I'd like to share some of our findings from the first approach, which is targeting aging mechanisms. Okay. So as Peter showed you from the hormones of aging, there's many different mechanisms of aging, so which mechanism to target? So our lab is particularly focusing on the epigenetic mechanism of aging. Okay. So let me explain. So this graph summarizes the life cycle of all the cells in our body. Okay. The cells in our body are generated from the stem cell. Okay. The stem cell grow and develop in young adult cells that form different organs. And that's the young organism that we all start with. And then many things happen to the cells and organisms, and eventually the cells become old and the organs become aged. And the aged cell are often associated with the parameters that Peter told you about: loss of function, loss of resilience, right? And many of these old cells are more prone to cancer induction, unfortunately. Okay. So this is considered as a natural process of what's the cells in our body going on. However, again, humans tend to change things. So a Nobel Prize discovery was made from the Yamanaka lab in 2006. So Dr. Yamanaka and his postdoc Takahashi identified that actually you can reverse this natural projection of the life cycle of the cells. You can make iPS cells, you can make stem cells out of the somatic cell. Okay. So you probably have heard of the power of stem cells therapy, right? You can differentiate the stem cells into all kinds of different cells. And those cells are young, and then you can use those cells to replace the damaged cell or the dead cell from our body, right? So that's one approach. However, this approach is quite lengthy, right? We have to generate these stem cells, and we have to differentiate them into young cells to replace the old cell. And also, many of our old cells, actually, we don't want to replace them. It's very hard to replace, right? How many cells we should replace and how to replace them is a very hard challenge, right? So we have an idea, actually, to, instead of replacing the old cells, we try to rejuvenate them, okay? Using the power of the Yamanaka factors, because this Yamanaka factor have this power to convert the young or old cell to the very young stem cells. Okay. So they do have the rejuvenation power, right? So what we try to do is use this rejuvenation power of the Yamanaka factor to make the old cells to behave like young cells. Okay, so this is an example of how we utilize this Yamanaka factors to rejuvenate the aged cell. So this particular experiment, what we did is we inject those Yamanaka factors into the retina of the mice, of old mice. So we use an assay. Basically, we play these moving stripes in front of the mice, right? So if these mice are young, they will be able to see this pattern. Because this pattern are moving, the mice will move their head together with the pattern, right? So you can see that the regular old mouse, they don't move their head too much because they lose vision, the retina is getting old, they lose the visual function, right? However, when we apply those rejuvenating factors, the Yamanaka factors, into the retina, these mice start to recognize this pattern again. Okay, so these are the same age. Okay, so because we only rejuvenated their eye, so unfortunately these mice still died at their expected age, but with a very healthy eye. So that's the thing. Can we rejuvenate the whole body? Right? Can we apply this concept to the whole body? That's something that we are actually working on. So specifically to talk about the topic today between aging and cancer, at SBP we believe the power of collaboration. Okay, so I don't have the cancer expertise. However, my mentor, Dr. Peter Adams, he has the power to test whether the cells can become cancer cells in the mice. Okay, so he showed that the old mice actually are more prone to oncogenic transformation to form those breast cancer. So by this natural collaboration, we want to test this idea. Okay, so again, the young cells will become old cells, and the old cells will be more prone to be cancer, right? And we want to apply this rejuvenation approach to make the old cells at least functionally like the young cells. And we want to test whether this rejuvenation approach will be able to resist this oncogenic transformation that naturally happens in these old mice. And we don't think this thing has to be natural. Okay, so again, humans tend to change things, and at SBP, we hope to change this dogma that the old cells have to become less functional and less resilient. Okay, so that's my end of the talk. I don't have data yet. We are still in the process of testing the data, and hopefully in 1 year, I will be able to share some exciting data. Yeah, so that's my end of talk, and I want to introduce Kay to talk about the clinical side, how we treat cancer, how to, particularly breast cancer.
All right, so thank you so much for the invitation. Thank you, Rima, for reviving this program. It's really exciting that the community comes together and really have a conversation. And today we're having a conversation about aging and breast cancer during the month of October. And thank you, Peter, for inviting me to give a talk and over the years of collaboration. So I just want to start off a little bit because I don't know if everybody knows me here. This is kind of redundant, but I started off interested in science, like many of the scientists here and probably many of you. And I went to Bronx High School of Science in New York, which is famous for producing a lot of Nobel laureates, so very smart people around me. And I fell in love, and during the same time, unfortunately, my father was diagnosed with colon cancer, and then I got involved in the American Cancer Society and actually was one of the summer interns to spearhead the screening mammogram drive locally in an Asian community where mammograms are a little bit underutilized at the time, and people don't want to talk about it. So that gave me a lot of experience and also exposure to somehow, I guess, naturally be more inclined in terms of chasing after the science as well as the clinical aspect of treating cancer. And this is where then it led me to go for schooling, and then at the same time being an emergency medical technician to kind of have the clinical exposure, which then led me to a 2-year stint as a research technician in the lab of Dr. Noah Noy, and studying the factors that can impact cancer, specifically the retinoic acid receptor, transcription factors. And unfortunately, my mentor, Dr. Noy, passed away from breast cancer. So that actually then really inspired me to really go after a MD PhD, and she was the inspiration for me to really go after that, and led me to NYU where I was in the lab of Dr. Herbert Samuels, again studying the process of apoptosis and particularly a special program in cell death in breast cancer. After that, I decided to come out west to see what's the fuss about after staying in New York all my life. And I stayed at UCSD for my residency fellowship. and postdoctoral fellowship at the lab of Jing Yang. As we mentioned, Jing has been collaborating with Peter, and it all really started with that. It kind of has started. So right now, I'm a breast medical oncologist, as the introduction has said. I work at the Coleman Comprehensive Breast Health Center. I'm a co-leader for the breast cancer clinical trial program, and I'm here to really talk about breast cancer. So want to start off with the collaborative research that Peter had already mentioned to really study the aging in breast cancer. So this is from what we call the SEER database, it's the public database of cancer. As you can see here, this pink line, I don't know if I can point, but anyways, it's very obvious. The pink line goes up in terms of cases of new breast cancer diagnosis as we move from age 0 all the way up to 70, and then just drop down a little bit for women. And then as you can see, for men can also have breast cancer, even though in a much lower incidence, that line kind of continues to go up. So as we heard, this study really, the collaboration really started between me, Jing, and Peter after a conversation at the C3 Council, so the Cancer Center Council, which is between UCSD, SBP, as well as Salk. And that's when Jing presented a new mouse model that we actually developed in her lab to study breast cancer at every single step, all the way from tumor genesis by introducing the oncogene of the cancer genes in mice, and then really be able to track the progression, so how the tumor grow, how it can spread, not just locally, but also to other areas like the lung. And using this model, we are able to do multiple things, including finding this particular finding, which is older mice, even though we expose them to the same amount of oncogenes, developed invasive breast cancer much more often than the younger mice. So this is really, we feel like this model recapitulated what we see in the world and clinically in our patients. And we can use this model as a way to really figure out the reasons behind this. And one of the reasons, as Peter had mentioned, is that maybe there's something about the cancer, the tumor cells, as well as the environment, that the aging process really maybe is a contributing factor. So here is a graph showing that there is less CD8 T cells, which are tumor-killing immune cells, in the old mice compared to the young mice. So in the young mice, in the tumor, is what we call hot tumor. The immune system is trying to fight it off. Whereas in the old mice, for some reason, the immune cells tend to stay back, relax, not care about it, or something that is not eliminating it. So this is why we potentially see a higher incidence of breast cancer development. So with this particular model, we are trying to test various existing medications that can potentially reverse this process. Now, we cannot test it in humans yet because we have to prove that it's actually working in a model before bringing it to humans safely. But I think this is a good jump start in terms of understanding what drives this progression, and then also having a platform, a way to test out therapeutics or medications that can reverse or rejuvenate the tumor cells. The immune cells. So with that, I want to go to the kind of big picture of how I think about working together in research, and how I think this collaboration locally between UCSD and SVP and other institutions nearby really help with improving breast cancer care. So first, we always have to start with the basics, the foundational knowledge of the cancer, how we can really prevent which is in this case, was we really want to prevent cancer. I rather prevent it than treat it. I always tell patients that, like, I'd rather not see you in clinic and see you do well outside of clinic. How we can pick up cancers early and how we can also have better treatment strategies. And then we move that research into the clinical space once we understand the foundational or the underlying mechanism and be able to test it in the clinical setting. And then we want to bring that to the population, so how we can actually deliver the care, how we can actually impact health outcomes by changing public strategies for really the greater good. So I think all of this is kind of like a cog in the wheel. We gotta work on all of them. So I'm really glad that here in this group we are really focusing on kind of combining both the basic approach as well as the clinical approach, and potentially bringing that into a clinical trial, to test some of the therapeutics. And then. The way I think about all of this is in this really complex healthcare system for breast cancer care is getting more and more complicated in a good way, because I think because of the fact that we are advancing our understanding of different kind of breast cancer, we're advancing our treatment of breast cancer, we're becoming more and more focused on patient-centered care. So patient-centered care, patients are in the middle. We start off with really understanding what they need, so what each patient needs in an individual basis, and that requires, having empathy, dignity, respect, because everyone's goal is not the same. And then sharing the information, so information sharing between not just the patient, but also their caregiver, the family, and then sharing between the clinicians and scientists who are trying to really change and advance care. And this really requires a lot of collaborations between not just the people I mentioned, but also with the industry, the philanthropy, the foundations, as well as governments to really support this, the payers and the healthcare system. So all of us has to participate in one of this, and in order to really engage everyone to really adapt to changing needs. So breast cancer care has been changing over the years for the better, and I would say ultimately really trying to measure the impact and value of all of this research, and then being able to bring it to the clinic and have patients in front of me saying, I am so glad that this is happening, that you're collaborating with the local scientists and researchers at SBP. So with that, I want to stop there and hand off this to the next person. Okay.
I think I just now, thank you everyone, thank you Kay. I think now I'll have Peter, Xiao, and Kay come up here and we'll do a Q&A session. And then, so have your questions ready. We'll have a microphone bringing around if start it off. Oh, okay, first question.
I'm in first, I can wait.
I'll go right after.
So you showed how the rates go up, and then as we age, they come down. Is anybody studying why those rates are coming down as we age?
That's a very good question, and you're thinking like a scientist. So yes, like why the extreme aged people, like the centenarians, they don't develop cancer anymore, right? So there's a statistical reason for that, because those people are already kind of like protected from because of the genetics, because of other factors, the lifestyle they utilize. Those people are the survivors, right? All of those people that died from cancer, from other diseases. So that's a statistical part. But biologically, yes, there are people that actually age slower and actually are protected more protected from all kinds of age-related diseases. And that's actually a very hot area to study how those centenarians can live so long healthily without developing all kinds of diseases. Right, exactly.
So 40%, I think at least 40% of the women in the United States have dense breasts. And they say the only way you can really tell is that they compare your old mammogram to your new mammogram because your present breast is so dense. So my question is, why, I mean, younger women have denser breasts, I know that, but why the older women still have the denser breasts that they had when they were younger? And why do some women have dense breasts and some women don't?
Very good question. Yes, yes. I wonder that myself too, and I do breast exam every day. I would say it's probably genetic and environmental factors. So women who are on, for example, exogenous estrogen, so estrogen or hormonal replacement therapy, for menopausal symptoms or other reasons, tend to have denser breasts. So we know that. But genetics also play a big part of this. And I would say nowadays the recommendation about dense breasts is that if you have a history of breast cancer and dense breasts, we do say that one should consider doing enhanced screening, so not just regular mammogram. And then when I say regular mammogram, I refer to the 3D with tomosynthesis mammograms, where you can actually have a higher sensitivity than your, the traditional mammogram. But right now, instead of that, you can consider doing MRI for detection. Now, MRI can lead to some higher rates of false positivity, so that leads to more biopsy, more worry about this new finding on the scan. So there's a balance. Now there's a newer technology called contrast enhanced mammogram that one can now consider for patients who have dense breasts. And that's basically just like what it means. You get an injection of contrast and then you do the 3D mammogram. That increases the sensitivity, not to the level of an MRI, but it increases the sensitivity quite a bit. So that's what I typically would utilize if we have an older woman who doesn't really have any other criteria for having an MRI, such as being a BRCA1 or 2 gene carrier, and they don't have a history of breast cancer, but I do worry about missing something, then a contrast enhanced mammogram would be the way to go.
So what's your feelings about the development of more widely available and easier screening procedures so that you get more people screened when they're younger? You want my mask up? More people screened when they're younger and perhaps capture the cancer when the T cells care.
Yeah, no, that's a very good question. So I think we've been struggling in the past to give general recommendations all across the board in terms of when to start screening. My position on that is to start screening at age 40, but obviously there are guidelines out there that starts at 50, and there are also guidance out there that says one should think about doing it every 2 years instead of every year. So I think this has to be done at, the recommendation or how to screen should really be discussed with the patient. The evidence should be presented, so pros and cons, because there are definitely cons in terms of screening when it can cause a lot of anxiety and discomfort, right? So it's a balance. I would say, yes, we tell all of our primary care providers to be on the lookout that if any of the younger patients report a lump or this abnormality in their breasts, not to go like, oh, this person is 35 and they can't have breast cancer. Really take it seriously and really get an imaging. I think that's probably the best way to catch any cancer development at the early stage when they're already, you know, when they can be cured.
Is any of your research, I know it's correlating a lot to aging and then the beginning of cancer, but does the aging or the research that you're doing also look into how the aging affects any possibility for the cancers becoming metastatic, especially with like metastatic breast cancer? Is that something that is also part of the research program?
Yeah, that's a really interesting question. We haven't looked at that, but Shan, who is sitting next to you, is the postdoc leading the study, and we have talked specifically about doing that because I think it would be really interesting to look at. But you know, Shan's fantastic, but can only do so much. So, but that's a project for the future, definitely.
Thank you. A lot of your questions stem around aging, but I'm wondering, is the research also being done by diversity, ethnicity, and also access to care? Because that is a contributing factor as well.
Oh, I'm nodding. Well, the reason why I'm not nodding is I hear you. So Dr. Angelique Richardson at UCSD is actually the breast medical oncologist who is very interested in health disparity, including biological and access type of disparity. So I would say the research is ongoing. I believe she was collaborating with someone at Sanford Burnham, but I think, yeah.
It's Swasti Harris.
Oh, yeah.
Yes, Swasti Harris Sharon had a specific interest in, I guess, breast cancer incidence and response to therapy across ethnicities. She had some kind of high-profile work published in that area. It was picked up by the BBC and other major outlets. Well, unfortunately, Swasti did move on a year or so ago to another institution. But so we have interests in that. But in my lab, I guess, I think it's a really important question. But that's given, I guess, the expertise and the tools that we use in my lab, primarily using mouse models, is obviously something that's very difficult. You can't really model it in mice. It needs to be something which is more looked at the human population level, which is what Swasti does. I can put you in touch with her if you'd like to discuss that with her more.
You mentioned about Yamanaka factor to use for retina. And recently, in 2024, there was a very breakthrough news through the group of Dr. David Sinclair.
That's my postdoc lab. Yes.
Oh, excellent.
Yes,
Yes.
I come from David Sinclair's lab. Yes.
Excellent. What they did, and they used all those knowledge, that factor, and they reversed complete aging in mice. They had mice, and in a few hours after whatever they did, all their hair color from dark went to white. They were very young. They got arthritis, I don't know, even age macular degeneration, something like that for their eyes, and in a few hours. Then they reversed it and they became young also. That means they were able to use that factor not just only in retina, in other parts also. And if you can, are you collaborating, which you said they are, collaborating with that team? Because it seems they are very ahead of reversing aging when it affects on arthritis and many problems
Right. Yeah, that's a fantastic idea. So, yes, I'm from the lab, and the research that I pointed out to is actually a collaboration from many lab members. Like it led by Yuan Chen Lu, who's a brilliant PhD student, right? So I was participating in that research. Yes, Yamanaka factor is considered as powerful regenerating factor, but they are also associated with potential risk of cancer. So that's some of the reason that we are hesitating to just directly utilizing them in humans yet. But David is actually testing that in the retina of the monkeys. Okay, so he has a company effort to do that, and he announced that there's a tremendous achievements from that effort that he can improve the vision from a disease model in the monkeys. Right? So translating to human is really hard. Because the safety concerns, the difference between human body and the mouse body, right? So we have to slowly target and test all those kind of possibilities. But yes, knockout factor, as what you said, has been proven to extend mouse lifespan. Okay. So that's been shown in independent research as well.
One other thing is, I was thinking, what will happen if they were able to reverse aging? Does this mean people, they don't die?
So so I often being asked about the ethical issues with what if we live forever, right? So that will cause a lot of eco-social system problems, and from my foreseeable future, I don't think people can live forever. Because what we are interested in SBP is to test the link between aging and diseases. Okay, so disease are our targets, right? But there's a lifespan limit, unfortunately, in almost all species as well. So there's a very strong limitation for human to live beyond 120, for example, because there's very few cases for human to live beyond that age. So that is kind of like a partially because of the genetics of humans. We just don't have those genes that can sustain us for living that long. I don't think there will be a close problem for us to deal with this ethical problems. Maybe in 1,000 years or 2,000 years, but nobody knows whether humans still exist.
Okay, one last question.
Peter, yeah, can I just add to that, because I think it's a really important point. So I think the way Xiao and I see it, we have a common view on this, that the goal of aging research is not really to make people live longer, but just to allow us to live healthier. Okay, so we're not trying to increase lifespan beyond the age of 80 or 100 years, but the idea is that instead of having living to 80 and having 20 years of chronic disease, which is obviously terrible for the person, for their family members, their loved ones, and also for society as a whole, because these people have to be supported. So not to have 20 years of chronic disease, but maybe just a few 2 or 3 years of chronic disease at the end of life. Okay, so we want people to live healthier, not longer.
Right.
One last question, and then we can have lots of questions outside during refreshment.
So I've been a participant in UCSD's breast cancer study for a lot of years, and I'm just wondering, have the results of that study contributed in any way to your research?
Which study are you referring to? UCSD has a lot of
breast cancer. So I fill out a questionnaire every year that's very lengthy, and they send me recommendations about mammograms and that kind of thing.
Okay. So this is probably the wisdom study
Yes,
you're referring to?
yes.
Okay, perfect,
Yes.
perfect. Yeah, so the wisdom study is a big part of UCSD's effort to really understand how we can prevent and screen patients who might be at a higher risk for breast cancer. And there are multiple publications that came out, and really using the data that's developed from that research, several of the screening guidelines have been kind of modified. And this is where we now more and more understand that the trends of breast cancer, unfortunately, is getting younger. So there are younger women now are being diagnosed with breast cancer. And then we also are developing preventative programs for patients who get, picked up on the wisdom study that don't have a genetic, like a true, what we call identified genetic risk factor, but have these potentially signal from their genes that are associated with increased risk. So I would love to connect, the wisdom study with the research here, especially since we have such a rich, network. The wisdom study is not only the UCSD, but it's, the multi-center across the country. So I would definitely think that this is a great idea to really connect the two. Yeah, there's a lot of information and data within the wisdom study. Yeah, thank you so much for bringing that up.
Thanks for all the great questions, everybody. And thank you to Zhao, Kate, and Peter. Please give them a round of applause. I think we had a great conversation, and let's continue the conversation over refreshments. But one announcement, Rima, is that I think in February we'll be talking about aging and neurodegenerative diseases. So please keep an eye out for that. So thank you again for joining us.